Hello everybody, I'd like to welcome you to our webinar today where we'll be going through enhancing performance through cell line replacement and process optimization. So before we begin, I'd like to let the panel introduce themselves.
Hello everyone, my name is Janet Lee. I am the Director of Cell Line Development Group. I'm really excited to be here today. Looking forward to have a productive discussion. Thanks for having me.
Hi, this is Sojeong Lee. I'm an upstream scientist. Nice to meet you all.
Hi, my name is Daniel Buckley, so I'm the Director of Process Development and Non-Clinical Production. Nice to meet everybody.
Hi, I'm Derrick Katayama. I'm a scientist in the formulation group.
So I think we'll dive straight in. So the first question that comes to mind for me is what are the challenges facing the biotech industry now, and how are Samsung meeting those challenges? So I might hand over to Janet to go through that.
It seems the quality and timelines are the most important factors in current environment. As regulatory bodies become more stringent, pharma companies are looking for contract development organization partner that has high quality standards with the right skill sets who could expedite their submission timeline at the first time submission.
I totally agree with that. And here in Samsung, we have a dedicated regulatory group, and that group has significant experience and a proven track record. Over the last number of years, they've over 240 successful submissions. In the last 2 years, we have over 32 successful IND submissions. So really, it is a very good history and a very successful story. And I might hand over to Derrick.
Yeah, no, I think one of the challenging aspects of the biopharmaceutical field is that it's very diverse and ever-changing. So, from new molecular modalities to new regulatory requirements that are there. And I think one way in which a biopharmaceutical company like a CDMO can compete in this is to have the personnel with the expertise and the experience. And Samsung Biologics does a great job of seeking out that global employees that have that experience or expertise. But they also have a working platform that is flexible, one that's modular, so it can adapt to the changing needs of the industry as well as the client.
Yeah, and I think that's key as well, and that contributes to the retention rate here in Samsung as well, having a flexible work environment, but also having a challenging and technical environment. So, and maintaining top talent, be it from Korea or be it external global expats, it's really, really key for the success of Samsung and for any CDMO. So yeah, I do agree with that, Derek. And then let's say there are a lot of challenges facing us, and how are Samsung evolving to actually face those challenges, Sojeong?
Oh, yes, then. In a very real sense, I think SBL is becoming a leading innovator in the CDMO field. So we have developed and implemented our manufacturing solutions to our process development and the clinical manufacturing as well. So this addresses some of the most pressing problems facing drug developers, which is our clients. And we have developed the specialized skills and expertise that are key for the successful development and manufacturing. Especially the complex molecules in the accelerated timelines and that are required today, but not very practical to implement in-house for our clients.
Yeah, and I'd agree. Look, based on complex molecules and the future modalities, and even the extra challenges that face the development of actually standard MABs and the accelerated timelines, there is a diversity of challenges. So I'd like to maybe go through some of the ways or some of the examples that Samsung have actually, I suppose, have developed to actually overcome those challenges, and maybe Derrick in formulation.
Yeah, no, absolutely. I think that there's a very challenging pathway to commercialization for any biopharmaceutical. And it becomes very important to be able to select the right sequence or molecule to move forward into development. And one criterion in which to be able to select that molecule or sequence is via a developability assessment. And Samsung Biologics offers a platform called Developability, which is a developability assessment that looks at the physical chemical properties of the molecule to help select that most appropriate molecule. And in doing so, the hope is that we set that program up for success from day one.
Yeah, and I really do like Developic because from a downstream point of view, it gives us our first insight into actually how purification runs with the molecule. So it's really beneficial for me because then I can actually scope out and make decisions based on actually initial data. So yeah, I really do like Developic. And I think obviously the key point for Developic is selecting the right sequence. And once you have selected the right sequence, we then move into cell line development. And I suppose I'd like maybe if you could go through the flexible options we have in cell line development here in Samsung.
So S-Choice CLD platform offers a flexible option to multiple programs for clients' development needs. Our S-Choice CLD offer standard process and transpose process. With our standard process, we have developed over 60% of monoclonal antibody stable cell line. Also bispecific, maybe 20%. Fusion and another 20%. So we bring in a wealth of experience across the modality utilizing our current platform. In addition, this year we have launched a transpose gene integration system. This is to really maximize the protein expression efficiency so the client has a lot of material to meet their sufficient clinical development needs. In terms of transpose gene integration, I would like to provide a little bit more insight. Transpose system allows stable gene integration. This is more of a semi-targeted gene integration system, allowing site-specific integration into active genomic loci. So the mechanism works by transposon and transpose, and by meeting those two, target site is targeted, so cut and paste mechanism leading to multiple insertion across the genomic integration, leading to very high productivity. So in our experience, monoclonal, we had achieved at least a 3 to 7-fold increase. Complex case, similar increase in productivity. In terms of the quality, purity is much, much higher utilizing transpose system, and also enhance the stability. So without the size limitation, the structural integrity is maintained utilizing transpose gene integration system. But in addition, we have more to tell. In terms of a timeline, everybody's very aware of accelerated timeline. Timeline for monoclonal, we shaved about 33% of a current standard timeline, leading to 2 months from transfection to MCB selection. For bispecific and complex, we shaved off about 25% of the development timeline, leaving about 2 months, 3 months for gene transfection to MCB selection. So we're very proud of this technology now.
Yeah, and I'd agree. This technology is a big step forward, and it definitely adds significant options to actually our cell line development capability. And as you said, we've seen significant improvements on purity, on titer, and on actually timeline. So it's actually, it hits everything. So that's it. Yeah, it's really key, and it forms a key cornerstone of our technology. So moving on, then, when you move from cell line development, next step is upstream. And how are these options impacting upstream? How do we integrate between cell line development and upstream?
Yes, then, needless to say, high quality cell is the most important factor for the successful development and manufacturing, especially from upstream point of view. So SBL Upstream has many options according to our clients' needs, which could be very flexible. For example, we can use the bulk pool for the early material generation for downstream formulation analytical method development. And this would add great values to expediting the timeline. But there's another strategy to minimize the quality difference between development and the manufacturing. We could suggest to use the single clone instead of the bulk pool or mini pool for both process development and non-clinical manufacturing. But even with this strategy, in a sense of reducing the timeline, our CLD has been working on various options such as transpose, she explained before, and of course, upstream and downstream side we've improved platform process development to be able to be used for the monoclonal antibody even without the development. This will help reduce the timeline in a whole sense.
Yeah, and let's say just listening there, some of the key words that are jumping out of me is quality and accelerated timeline. And then actually a question that jumps into my mind, it goes back to develop pick, which is actually key for accelerating our timeline and getting an early indicator and selecting the right sequence. So then what pops into my head is, how do we make sure that the material that we generate is representative to actually do the testing and develop pick? So maybe Janet, would you go through that?
We have an answer for that, Dan. So our S-Choice transient expression system was successfully generated utilizing high-quality transient expression material which enables the early material supply needs for our client needs. So what S-Choice did was during our development, we have tested across the modality from monoclonal to complex to FC fusion. In terms of the productivity, we have achieved over gram per liter quantity within very relatively fast timeline, very comparable to a commercial platform that's available to our clients. In terms of the quality comparison, based on percent purity analysis, which is an intact formation, the percentages are so high, very comparable to existing commercial platform, which enables thermal stress performance analysis, which is done by Developability, and enabling a lot of options for our development clients' development needs.
Yeah, and like even just taking a step back from the conversation so far, it's very evident to me that cell line development is actually key for the success of any molecule. So then another question that comes straight to my mind is actually, what should you consider when you're selecting a strategic partner with regard to cell line development? And I might hand that over to Sojeong.
Oh, yes, then. I think everyone knows that only the first and the second entry drugs can take the market share. And that's why our drug developing clients want the fast timeline as much as possible, especially for IND. No wonder why the robust and highly productive cell line generation can assure the expedited timeline, because the smart cell line is the very starting point of the journey of the molecule. So in addition to the cell line development, I think the robust platform process would be a great benefit that adds value. When a strong cell line combines to the robust platform process, this will greatly have impact on COG, cost of goods, because you could get maximized effect, such as the reduced number of batches and reduced risk in batch failures. So these two horses, robust cell and process, these are the two key for the successful development manufacturing. And I think last but not least, the mindset of the employees, such that how much they are flexible to the client's needs and how much they preemptively prepare for the quality in mind is going to be another key.
Yeah, best in class or first to market is key. But one of the other considerations that I think is very important is actually cost of goods. Like over the life cycle of a product, that is actually key. And as you said, reducing batches, reducing costs, etc. But what I find amazing is somebody in development already thinking about the commercial side, already thinking about the cost of goods. And it reminds me of one of the key advantages here in Samsung, and that's end-to-end on one site, and where everything is co-located. So maybe I might hand over to Derrick and maybe get your opinion on actually that key advantage here in Samsung.
Yeah, no, absolutely. I think it is a key advantage to have everything on one site, from early development to commercial manufacturing. I think that the main advantage for that is seamless communication. The early development labs are just walking distance away from the MSAT groups, and so communication is facilitated by having a face-to-face meeting. And another thing that's really beneficial is that the early development labs and the scientists working there understand the facilities in the manufacturing, so they understand the capabilities and the limitations for the processes like buffer makeup or drug product filling.
Yeah, and I totally agree with you. And actually in Samsung, we're even taking a step further where we're moving people from MSAT into the development area and from development into the MSAT area. So that means then, as you said, we're fully aware of the technical capability of the equipment and the technology in our commercial side of manufacturing, which means then during development we take into consideration, and we can avoid any significant capex or any delays in late stage commercialization. And then again, one of the key things that pops into my mind is in upstream and scalability, and what would be the key benefits for end to end, all on one site here in Samsung from an upstream scalability point of view?
Yeah, so like Derek and Dan, you just mentioned, I think the facility fit is one of the key factors for the scalability. So of course, we can match our processes to the manufacturing, whichever manufacturing fits are. But in SPL cases, and in my experience as an upstream scientist, we develop our processes according to the larger scale manufacturing sites at SPL sites that could be easily transferred to the MSET or any manufacturing facilities at our Samsung Bio campus. So I think, imagine even within a single company, the same company, if the location of the process development and the manufacturing is different, maybe the communication will be a hassle. So maybe Zoom call, it's not very visible every time. So I think walking distance between the development and the MSET manufacturing, that'll help a lot in a sense of the scalability, because we can talk every day very easily about the process development strategies and schemes to be able to fit the manufacturing scales.
Yeah, and I see it even within analytical, from site to site, you can see variability within analytical, but when it's all in one site and we're using all the same equipment and the same training, etc., it reduces the risk of variability even within analytical. So it's a benefit across every single function. I think we're probably coming to the most exciting part of the actually webinar, and I know it's the part where Janet is the most, let's say, most excited about, and actually really happy about the success stories we've had in cell line development recently. So I might hand over Janet to go through those success stories.
Sure, thanks, Jen. So I guess this is the most exciting part of this webinar, because we had a success story not from cell line development group alone, but this is our CDMO organization. Just wanted to able to share the showcase of this experience we recently had. We had clients low expressing CHO cell line, which was IgG4. We have developed the cell line from very low expressing to reasonably high, relatively high expression level, utilizing efficient S-choice CLD platform to improve overall productivity, but still we have to meet the target CQA. So it's a challenging project, but we work as a team. So whether it's a cell line development project or entire project, we work together in a matrix organization. So from the beginning, we have invited upstream, downstream SMEs, including PP formulation, and we worry about how do we best improve this molecule for clients' best timeline. So in this case, we have applied CLD, S-choice CLD platform, and the target molecule was expressing about gram per liter, low expressing. Upon improvement, we were able to achieve at least 2.4-fold. So we were very excited with this outcome. However, when we run the quality analysis, the target CQA, including the charge variants, were slightly outside of the window. So we tossed this task to Upstream, and Sojeong's group took care of it. Sojeong?
So thanks to CLD and analytical group, Upstream quickly gets an idea on quality items, how to control during the cell line development stage from their quality analysis results. Mostly, Upstream uses either process parameters such as temperature, pH, or media supplements for the quality item control. So we have a list of parameter candidates or component lists for certain quality aspects such as glycosylation, silylation, charge variant, and other quality aspects. So followed by the design of experiment, verification in 2-liter scale bioreactors will tell us whether the process shows the comparability to the pre-existing processes. So then, how did the downstream process development go afterwards?
Yeah, like with this project, obviously it was a cell line replacement project. So the client already had a downstream process, and they already had identified the critical quality attributes, the operating process parameters, etc. So the first thing we did was actually, once we got material from the new cell line and from Upstream, we did a feasibility run and we checked, look, what are the areas that we need to do some optimization in, and some development in, to ensure purity, to ensure good yields, optimum yields, but also to ensure comparability at the drug substance phase. And what was key for us was actually understanding the critical quality attributes and making sure we had all the right analytical methods, and that we were actually, let's say, we understood where we needed to actually do our development work. And it turned out very successful because, again, we had the right analytical methods, and then we were able to actually use the appropriate purification unit operations to achieve what we needed to with regard to comparability for drug substance. And that's what's key. What's really key for all projects is do the correct screening as early as possible, so then we can adapt the downstream processes to deliver the drug substance at the right quality, at the right yield. So yeah, a very, very successful project. And as Janice said, I think everybody across the whole organization was really proud of that, of the achievement associated with that project.
Yeah, so to conclude the case studies, successful CDO team development network together, we were able to achieve overall 3.4-fold increase from the start to finish, while we have hit the nail on the verification of the CQA. Very successful case studies, and client was very happy. Case B was just cell line development project. This one was really low expressing, DHFR expression platform, very old cell line. We were able to achieve approximately 9-fold increase upon efficiency of the platform using S-choice. So this is another happy story. Case C was already decent cell line expressing over between 2 to 3 gram per liter. Upon CLD platform implementation, we were able to double the CLD productivity. So overall, this was a very happy experience for our group, and we encourage our client to try cell line replacement option for their development needs.
Yeah, and as you said, all these options are showing, let's say, a very successful track record here in Samsung Biologics. So it's something that we're very proud of. I think the time has gone very, very fast, unbelievably fast in my mind here. So I think maybe to finish up, what we might go through is we go through each panel's opinion on why Samsung Biologics would be the correct strategic partner for biotech companies out there. So maybe I hand over to Janet again first.
So as mentioned, I think we have really new ways of working in Samsung Biologics. Not only we work with the clients of development limited scope, we really want to be your CDMO partner, enhancing our capability and really leverage our large-scale manufacturing capability in our site. So we really work together as one team. We want to be really extended partnership of your development needs, so that we incorporate cell line development, upstream, downstream, analytical, and DP formulation to really think ahead and really support clients' accelerated time need with the right success to their development requirement to get to the patient faster.
Yeah, Janet, like I totally agree with actually those advantages, because over the last few years I've been working in those teams, and I definitely feel that the cross-functionality associated with that matrix approach actually adds and definitely ensures that we reach, let's say, we reach optimal solutions when we encounter challenges, but it also means that actually that cross-functionality and that team actually operates most efficiently. So yeah, I think it's a big advantage the way we actually operate here in Samsung. And maybe just on the same topic, Sojeong?
Yeah, I think we have to think about the key factors that must be addressed. Regulatory, sustainability, confidentiality, quality, speed, and cost. So Samsung believes that a right first time approach results in high quality, which in turn enables faster development timelines and reduced costs, while still ensuring sustainability, confidentiality, and the regulatory compliance. So because we have a flexible development approach built into our operations as well, we were able to execute the right studies at the right moment and using the right analytical methods to ensure that we identify and address any development challenges that may arise as early as possible.
Yeah, right first time is a key philosophy, and it's something that actually I fully believe in, and it's something that it's key, as you said, from a cost point of view, from product quality point of view and from a schedule point of view. So yeah, no, it's really important. And maybe Derrick, in your front.
Yeah, no, absolutely. I think, as a scientist in the field of biopharmaceutical industry, I do my best to try and stay on top of what's coming up on the horizon. And I feel like it's incumbent upon the leaders in the biopharmaceutical industry to also invest into the future. And in my short time here at Samsung Biologics, I've seen them invest in the future with these proprietary platforms that we talked about today, as well as the investment into new modalities like antibody drug conjugates. And with regards to scale, in my short time here, I've seen the scale increase tremendously. In fact, if I look across the street, I see a plant that's being built that's going to have 180,000 liter capacity, which is tremendous. And this makes me excited for the future that that will enable Samsung Biologics to continue to make medicines, these transformative medicines, to patients across the globe.
Yeah, no, and it's something that's blown my mind away as well. When I came here first and I saw how fast everything had expanded and how successful, even during rapid expansion, how successful everybody was and how actually successful Samsung was. And then over the last 2 years, I've seen plant 4 come online. I've seen an ADC plant starting being built. I've seen a brand new biotech plant just starting to be finished in 2025. And I've seen new labs in CDO being built and successfully integrated into the systems. It's just how fast and new modalities coming online. It's just the speed and how successful everything is integrated. Yeah, and it fills me with excitement for the future of Samsung Biologics, but also fills me with a lot of confidence with regard to how successful we have been in the past in actually bringing all these new capabilities online. So I think at this point we might wrap up because we have run out of time. But look, on behalf of the panel here, I'd like to thank everybody who tuned into this webinar, and I hope to see you soon in the future. Thank you, everybody.